This is the structure of both cilia and flagella. There are three different regions of both cilia and flagella. They are exonym, transitional zone, and basal body. Cilia and flagella are assembled from a basal body. Basal body consists of a disc-like structure. The transverse section of this disc-like structure looks like this. If we will take the transverse section of basal body, then it will look like this. From this transverse section, we can see that basal body contains 9 triplet microtubules. The transverse section of this region of basal body generally looks like this. This transverse section consists of 9 linked triplet microtubules. If we will take a closer look at any one linked triplet microtubule, then it will look like this. Each linked triplet microtubule consists of A tubule, B tubule, and C tubule. The transitional zone is present between the basal body and exonym. If we will take the transverse section of transitional zone, then it will look like this. If we will take the transverse section of exonym, then it will look like this. Exonym is the membrane-bound core of both cilium and flagellum. The transverse section of exonym consists of 9 peripheral doublet microtubules. Each doublet microtubule in a closer look generally looks like this. This doublet microtubule consists of A tubule and B tubule. The A and B tubules of basal body are continuous with the A and B tubules of the exonym. This is the diagram of transverse section of a cilium with its plasma membrane. The transverse section of cilium consists of central pair of singlet microtubules and peripheral 9 doublet microtubules. Each doublet microtubule consists of A tubule and B tubule. A tubule has 13 protofilaments and B tubule has 10 protofilaments. The major motor protein present in cilia is axonemal dynein. Axonemal dynein is a large multi-subunit protein. Axonemal dynein is related to cytoplasmic dynein. 2 rows of dynein motors are attached periodically down the length of each A tubule of the outer doublet microtubules. These dynein motors are called as the inner arm dynein and outer arm dynein. Inner arm dynein and outer arm dynein interact with adjacent B tubule. This adjacent B tubule bring about bending of cilia and flagella. The central pair of singlet microtubules is connected by periodic bridges like rungs on a ladder. Nexin protein joins adjacent outer doublet microtubules. Radial spokes project from each A tubule of the outer doublet microtubules towards the central pair of singlet microtubules. This is a radial spoke head. Now some important points. The length of cilia and flagella range from a few micrometers to more than 2 mm for some insect sperm flagella. All the microtubules in cilia and flagella have the same polarity, that is, the plus ends are located at the tip. Please like, subscribe and share.